the hollow portion. Is everybody kind of with me? Okay? Because if we think about the intestines, it's our foodstuffs that are moving through it. Okay? So there is an area that's hollow. So anything that meets that hollow point is a free surface of that cell. Is everybody with me? Okay? Because if this were the outer portion of this tube, okay? So if this were the outer portion of it, there would be cells all along it, but then I have the hollow portion that's allowing the foodstuffs to move, okay? So I end up with those microvilli, the little finger-like projections, and they're, in some cases, because they do have a free surface, they can have cilia. Hair-like projections, do you guys remember that from cells? Okay. So what they do is they're giving a huge amount of cell plasma membrane available for absorption, and then these cilia, because they act in a wave-like fashion, continue to help propel the substances through, for example, the intestines. And this portion right up here is considered the free surface. Is everybody with me on that? Okay. So those cilia help to move materials across. We can have in the epithelia foldings that take place. Like I mentioned, if the organ needs to be able to change shape, i.e., for example, the bladder, it folds flat and it'll look crinkly, so to speak, okay? And then as you drink the fluids fill into it, it begins to fill up for the bladder. Our stomach, in between meals, goes a little, not completely flat, that would be bad, okay? But it will kind of flatten a little bit. But if you've ever seen pictures of inside the stomach, it's very crinkly. And it allows for expansion when we eat, okay? So, epithelia are good for that to take place. So, this is another one of the videos, and it's probably going to get into, John, what we're going to get into about these exchanges of material and so forth, but it'll give you an idea of these categories of epithelia.
Epithelia form the surface layer of the body, line body cavities, hollow organs, and structures, and constitute most gland tissue. Any substance that enters or leaves the body must cross an epithelium. All epithelia consist of one or more layers of tightly packed cells and little or no extracellular matrix between cells. Distinguishing features of an epithelium include a free apical surface and a fixed basal surface. Specialized intercellular junctions join adjacent cells, and a thin basement membrane separates the basal surface from underlying connective tissue layers. Although epithelia lack blood vessels, many sensory nerve endings are present. Epithelia have a high regenerative capacity that allows them to maintain their function. The primary functions of an epithelium are to resist dehydration and injury from physical, chemical, and biological agents, to selectively regulate materials entering and leaving the body, to secrete products produced by epithelial glands, and to monitor the environment through specialized sensory nerve endings and specialized epithelial cells. Epithelial tissues are classified using two basic criteria: the number of cell layers and the shape of the cell at the apical surface. An epithelium composed of one layer of cells is termed simple. If there are two or more layers of cells, the epithelium is termed stratified. When the apical cells are flat and thin, the epithelium is classified as squamous. Apical cells that are somewhat square-shaped belong to a cuboidal classification, while those with a rectangular shape are termed columnar. By combining the criteria of cell shape and number of layers, all epithelia can be classified easily. For example, a simple squamous epithelium is composed of a single layer of thin, flat cells. In contrast, a stratified squamous epithelium is composed of multiple layers of cells, with the apical surface layer being thin and flat. These same tenets could be applied to cuboidal and columnar epithelia. There are two special types of epithelia. Transitional epithelium, found in urinary tract, is a stratified epithelium that gives the impression of transitioning from cuboidal when the epithelium is relaxed to squamous when it is stretched. A pseudostratified epithelium, found in the trachea, for example, is a simple epithelium in which all cells contact the basal lamina, but only some reach the apical surface. Cells are of varying heights, and their nuclei are at different positions in the epithelium, giving the impression of stratification. Some epithelial classes have specializations of the free surface of apical cells. Simple columnar epithelial cells in some organs have cilia, which are small, finger-like motile projections. Thus, simple columnar epithelium is subclassified as ciliated or non-ciliated. In parts of the digestive and urinary tracts, epithelial cells involved in absorption have a dense collection of short cytoplasmic processes called microvilli on their free surface. Microvilli are non-motile and serve to increase surface area. Because the microvilli appear fuzzy in the light microscope, cells with numerous microvilli are said to have a brush border. A stratified squamous epithelium has either dead or living cells as the surface layer. The dead cells contain the protein keratin. Therefore, stratified squamous epithelium is subclassified as keratinized or non-keratinized. Epithelial cells are tightly connected to adjacent cells by specializations called intercellular junctions. There are four types of junctions: tight junctions, adhering junctions, desmosomes, and gap junctions. Tight junctions, also known as zonula occludens, are bands of fused cell membrane near the free surface of adjacent epithelial cells. These junctions attach a cell to its neighbors, encircling cells and sealing the space between them. Thus, substances crossing the epithelium must pass through epithelial cells, not between them. Tight junctions are essential to the primary function of an epithelium, which is to regulate substances that enter and leave the body. Adhering junctions, also known as zonula adherens, have a similar band-like arrangement seen in tight junctions. They are located deep to the tight junctions and have numerous microfilament proteins from the cytoplasm that act as anchors for adjacent cells. There is a small gap between the cell membranes of adjacent cells at adhering junctions. Desmosomes, also known as macula adherens, are small button or snap-like junctions. These form at the points of mechanical stress. They are different from tight and adhering junctions in that they do not encircle the entire cell. Gap junctions are points where small pores connect the cytoplasm of adjacent cells. The gap junction consists of a series of hollow bridging structures called connexons. Small solutes, such as ions, glucose, and amino acids, pass through connexons from the cytoplasm of one cell into a neighboring cell.
Okay, so that was a really good representation of what we've talked about, except for those ways that the cells connect together. We haven't gotten to that yet. So, keeping this information in mind, all right, if we now look,
Epithelia form the surface layer of.
there we go. So now, if we look at these cells that exist and going to each one, simple squamous tells me one layer of the fried eggs. That's what it looks like. I mean, you know, if you look at them, they look like little tiny fried eggs, okay? Because the nucleus is kind of big, and it actually pokes out a little bit, okay? These are the areas of the body we find it. So, we find it lining of blood and lymph vessels. So think about your blood vessels, lymphatic vessels. Small ducts, DUCTs, where a substance needs to be released from. In the alveoli of the lungs. Because one of the things that this simple squamous structure was very good for, diffusion of gases, where I need oxygen and CO2 to move. The loop of Henle and the kidney tubes, which you'll get into in part 2, all right? But if the kidneys are for getting rid of waste, this structure is great for that. Lining of serous membranes and the inner surface of the eardrum. Their function: diffusion, filtration, protection against friction, secretion, absorption. Very great for the moving of materials. One layer of the squamous cells, one layer of the fried eggs. Simple cuboidal, one layer of the cubes. We find this: kidney tubes, glands and their ducts, for example, oil glands, sweat glands. The choroid plexus of the brain. This is the area of the brain that gives rise to cerebrospinal fluid. You'll get into that in part 2 also. Lining of terminal bronchioles, meaning the point where you're going to be reaching alveoli. And the surface of the ovaries. Their structure, the single layer of cubes. Some can have microvilli. You find that in the kidney tubes. And some could have cilia, the terminal bronchiole of the lungs. Their function: secretion and absorption, very good in the kidneys. Secretion in the glands, like we'll look at in a few minutes. The choroid plexus of the brain for secreting or helping to form the CSF. Movement of mucus in those terminal bronchioles, because that is the point where if you have anything trying to get into your respiratory system, all right, you want it, you don't want it, actually you don't want anything making it much past here, okay, where the bronchi branch. But if any pollutant or anything like that makes its way past, the body is still trying at the point of the alveoli to get rid of it. It does not want it in the alveoli at all in the lungs, but you'll get into that part 2. Simple columnar, a single layer of the columns found in glands, ducts, some of them, bronchioles of the lung, auditory tubes, uterus, uterine tubes, stomach, intestines, gallbladder,